So I think what we are appreciating very much more is that to give a diagnostic label to a condition is perhaps not the correct thing to do, in that there are very subtle gradations in terms of subcategories within that diagnostic label. And very much what we're now looking to do is to work out exactly what Mark and Melita and Paul have been alluding to, is to work out what is the appropriate diagnostic label that I apply to this person. How do I understand the fine gradations of the way that that disease is likely to behave, and what is the best treatment that I can pick for that person based on the way that I expect that the disease will behave in response to different therapies. So it's moving towards not only understanding how the disease behaves, but how it's likely to respond to the many therapies that we have in our armamentarium, but also looking towards, can we design treatments that then pick on those particular weaknesses that the disease may develop.
So is that happening now?
I believe it is, and certainly the basic research that we're doing, that Mark does in the lab and that all of us do in the lab, looks at understanding the fine particularities of those fine gradations, breaking the disease down into more subcategories. But also something that Melita alluded to is that looking back on the clinical trials that we've done, looking back on the information that we have about patients who've been in clinical trials, and understanding why a particular subgroup, and as you say, the tail that wags the dog, picking out what the factors are with those patients and why they respond to one treatment, another patient may not.